going to start with our today's session, guys. All are irreversible steps in glycolysis except one: phosphofructokinase, pyruvate kinase, and then the enolase, and then, my dear friends, hexokinase. Okay, so the poll begins right now, and you are even having the option of poll here right now, guys. So let me see how many people will answer correctly, and let me see, who will answer very fast. Yes, my dear friends, most of the students are answering the correct answer, almost like 64%. 64% people are answering the correct one. Okay, sir, fine. So now, my dear friends, as I told you even yesterday also, yes, as I even told you yesterday also, we are not at all concerned about the questions there, guys. What we are concerned here right now about the concept. Okay, na? So just with the help of the questions, I'm discussing with the entire concept. So exactly same like our yesterday's class. Let's get started with our today's class, guys. So be ready with your pen, pencil, paper, and all those, guys. Whatever I'm telling you, just mark my words there. Whatever I'm telling you, just mark my words there, guys. Okay, na? So let's get started right now, guys, with that same energy now. So come on, guys. So now in this question, I'm actually going to tell you all the points related to glycolysis. Okay, na? Bhai ye OP kya hai yaar? OP. हाँ जी. So let me teach you all the points which are important about glycolysis. Welcome. Now, now, my dear friends, yes, what is the first thing to learn here right now? Okay, what is the another name of glycolysis? Yes, what is the another name of glycolysis? It is EMP. And remember, EMP stands for what? It stands for Embden-Meyerhof pathway. So, सबसे पहले ना उसका नाम सीख लो. Another name of that one is Embden-Meyerhof pathway. And after learning the name of that, what else they can ask you in the exam, guys? I hope everyone knows the definition of glycolysis. In glycolysis, what is going to happen? We are going to convert the glucose molecule to pyruvate. Okay. And what is the end product of aerobic glycolysis? If it is aerobic, the end product will be pyruvate. And remember, if it is anaerobic glycolysis, the end product will be lactate. Okay. Guys, if you have even attended my regular classes, and all, even then I request all of you to please take a pen, paper, and start here, guys. Points you have to refer before going to the exam on 31st morning. Is it okay? So welcome here. So glucose will be actually getting converted to pyruvate, the end product of aerobic glycolysis. And what is the end product of anaerobic glycolysis? That will be your lactate. Lovely. Now, what is the second question that they can ask you here? Where is glycolysis taking place? What is the location of glycolysis? Glycolysis will be taking place in cytosol. Every student knows that. The third thing that they can ask you here is the rate limiting enzyme of the glycolysis, where we can regulate, where we can control glycolysis. And I think most of the students will be knowing this one. That is your PFK1. And what is the full form of the PFK1, sir? That is nothing but phosphofructokinase 1. Phosphofructokinase 1. Come on, guys. Start like the same thing. Same like our yesterday's class here right now, my dear friends. Don't get distracted. For next 1 hour, you people are like completely along with me, guys. Forget about everything in your life. Okay, now forget. Sab kuch bhul jao, sir. 1 ghanta. Yes. Our important 17 marks of biochemistry. Yes. The moment I tell you the rate limiting enzyme, repeat that rate limiting enzyme in your mind, guys. Is it okay? Fine here. What is the rate limiting enzyme, sir? Phosphofructokinase 1. Phosphofructokinase 1. PFK1. Okay. And after the rate limiting enzyme, what else they can ask you? Yes. What is the net gain of ATP in what is the net gain of ATP in glycolysis, guys? Yes, if it is aerobic glycolysis, the net gain of ATP will be like 7 ATPs. Okay. And if it is anaerobic glycolysis, remember the net gain of ATPs will be 2 ATPs, guys. So the net gain of ATPs in aerobic glycolysis will be 7 ATPs. In anaerobic glycolysis, it will be 2 ATPs, guys. Lovely. I'm seeing all your answers there. Priyanka, Ravi, Shruti, all of them are answering perfectly correct there, guys. Yes. End product of aerobic glycolysis, 7 ATPs. Anaerobic glycolysis will be like 2 ATPs, guys. Lovely. And after that, my dear friends, the next important thing is glycolysis reversible or irreversible, guys? Can you please answer me? Glycolysis is reversible or irreversible? Remember, glycolysis will be like irreversible. And we are having like 3 reactions, like irreversible reactions in glycolysis. So please write down In total, there are like 10 reactions in glycolysis, and out of the 10 reactions, 3 are irreversible steps. And I always tell my students to remember like this in my classes. Number one, the first one, hexokinase and glucokinase. And the third enzyme, guys, that is your phosphofructokinase 1. And now the last and final one, that is your pyruvate kinase. I hope you all know the full forms of that one. Okay, so I always ask the students to remember, just like a song there, guys. HK, GK, PFK1 and PK. चलिए मेरे साथ में बोलिए सर सब लोग जोर से बोलो. HK, GK, PFK1 and PK. HK, GK, PFK1 and PK. Hexokinase, glucokinase, and then phosphofructokinase, and then the pyruvate kinase, guys. Okay. Then आपको enzymes के नाम बोलना है, ठीक है ना? हाँ जी, जोर से बोलो. Hexokinase and glucokinase, phosphofructokinase 1 and pyruvate kinase. These three are the irreversible reactions of glycolysis. Is it okay, बच्चों? That's all. Okay, so whatever points that you are able to see on the screen there, these are the important points. Along with that, you add one more point here right now, sir. लिखो वहां पर inhibitor. Yes, do we have any inhibitor of glycolysis? Of course, you have the inhibitor. What is the name of that? Fluoride. Fluoride. Sir, ये fluoride किसको inhibit करेगा? It is going to inhibit the enzyme enolase. Which enzyme? Enolase. लेकिन यार मैं एक बात बताऊं आपको. While studying in the class, it looks very nice. Okay, sir. Very good, sir. What is the inhibitor, sir? Fluoride, sir. But in the exam, they will never give you like that. बड़े प्यार से वो आपको ना fluoride करके नहीं देगा. In exam, always it will be given in the form of NaF. And what is NaF? Sodium fluoride. Sodium को मारो गोली. Your concentration should be on fluoride. आपकी नजर ना fluoride पर होनी चाहिए. Fluoride is gonna inhibit what? Enolase. Class. ठीक है? So yes. Now today we will promise to ourselves on 31st August 2020 morning, I'm going to see these points and go. Okay, come on. Number one, what is the another name of glycolysis? Embden-Meyerhof pathway. Definition everyone knows. Aerobic glycolysis, end product pyruvate. Anaerobic irreversible reaction HK, GK, PFK1 and PK. And finally, the inhibitor will be the fluoride. That's all. Now, after learning these many points, guys, let them ask anything. We are ready for that one. Understanding here right now? Let them ask anything in glycolysis topic. We are ready. So welcome back to this question here right now. Phosphofructokinase, pyruvate kinase, and hexokinase all are irreversible except for enolase. So welcome back to my class today, guys. The first concept completed, guys. Done. So with the help of the questions, we are going to do the entire concept, guys. Lovely. Okay, na? Seeing you all back today. आज ना और भी energy ज़्यादा है. Welcome, sir. Welcome to the second concept here right now. Which of the following enzyme helps in production of NADH? Helps in production of NADH in citric acid cycle. Lovely. Forget about question, sir. Question ko maro goli. Welcome to the second concept here right now. That is citric acid cycle. Yesterday it was all about proteins, and today it's all about the carbohydrates, sir. Welcome, welcome, welcome to the next topic. Okay, sir. Krebs cycle. What is another name of the Krebs cycle? Come on, sir. Thoda sa na fast, so that we can cover many concepts. Krebs cycle is also known as citric acid cycle. It is also known as TCA cycle. TCA cycle, guys. What is the full form of TCA cycle? Tricarboxylic acid cycle. ये तो आपको पता ही होगा. Now, first thing that you have to remember is the location. Where is Krebs cycle taking place? Mitochondria. And where inside the mitochondria? The matrix. Mitochondrial matrix. After that, what is the rate limiting enzyme of the Krebs cycle? It is IDH. What is IDH? Isocitrate dehydrogenase. What's the rate limiting enzyme of the Krebs cycle, sir? Isocitrate dehydrogenase. Dehydrogenase. Okay, bachcho. Now, after the rate limiting enzyme, the next thing, net gain of ATP. What is the net gain of ATP in Krebs cycle? Remember, totally 10 ATPs are produced in Krebs cycle. But remember, from each molecule of acetyl CoA, we are going to begin with acetyl CoA combining with oxaloacetate. Remember, whenever you begin with one molecule of acetyl CoA, at the end you'll be getting 10 ATPs. Lovely. And now the oxidation reactions, guys. How many oxidation reactions do you have in Krebs cycle? In Krebs cycle, you'll be having totally 4 oxidation reaction. Totally how many, sir? 4 oxidation reaction. And what are those reactions? Number one, One will be your isocitrate dehydrogenase. Another one will be alpha ketoglutarate dehydrogenase. Another one will be succinate dehydrogenase. And the fourth one will be the malate dehydrogenase. You can see the dehydrogenase. D means a removal, and a removal of hydrogen is nothing but oxidation. A removal of hydrogen is oxidation, guys. Okay. So how many oxidation reactions do you have in Krebs cycle? There are like 4 oxidation reactions here. Okay. Isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinate dehydrogenase, and malate dehydrogenase. Remember, in all of them, what is going to happen, guys? There'll be formation of NADH. In this oxidation reaction, there will be formation of NADH plus H plus. And alpha ketoglutarate, yes, NADH plus D. But remember, succinate dehydrogenase. Here, there'll be formation of FADH2. And again, malate dehydrogenase, the same thing. In this also, what is going to happen? So NADH plus H plus is going to be produced during that oxidation reaction. I think now at this point of time, my dear friends, another 4-5 days left out for the exam. I think now you already know what is oxidation. Now don't ask me to go to that school days basics right now. Okay, sir, oxidation क्या होता है? ठीक है ना? वरना गड़बड़ी हो जाएगी. Oxidation means removal of hydrogen, and the name of the enzyme is dehydrogenase. And in biochemistry, guys, that hydrogen is accepted by either NAD or FAD. So at 3 places you are getting NADH, at 1 place you are getting FADH. Okay. If something is striking in your mind, yes, if something is striking in your mind, guys, I can tell you that you recall my yesterday's class. In my yesterday's class, I taught you about electron transport chain. Let me see. Can anyone tell me what is the name of the complex number 2? Complex number 2 will be nothing but your, yes, the succinate one, sir. Succinate dehydrogenase, and their FADH2 is the one which is contributing the hydrogens there, guys. Lovely. So now on the day of the exam, these are all the points you are going to recall for the Krebs cycle, sir. That's all done. Always remember the another names of that, where it is going to take place, the rate limiting enzyme, net gain of ATP, and the oxidation reactions, and then you are done, guys. Welcome back. Enzyme helps in the production. यहाँ पर प्रोड्यूस होगा सक्सिनेट थायोकाइनेस, नो. सक्सिनेट डिहाइड्रोजनेस, it is FADH, no. Isocitrate dehydrogenase, yes, it is NADH. And fumarase, no. So NADH in citric acid cycle is produced in the third enzyme. The points which are required for the MCA exam, sir. That's all. These are the golden points. Okay, now. And Veer is asking the what is the difference between NADH and NADPH? Veer, the answer is in your question itself. Yes, NADPH is actually the phosphorylated form. NADPH will be actually the phosphorylated form there, guys. Welcome. Next one. Welcome to the next one, guys. Okay. Enzyme deficiency seen in the Pompe's disease. What is the enzyme deficiency seen in the Pompe's disease? Oh, ho. Now, this is one of the hot favorite topic of your MCI examiner. So I think you are automatically understanding here. I'm slowly moving towards where, guys? I'm slowly moving towards the, yes, the glycogen storage diseases. समझ आ रहे हैं? तो आज के लिए सर, in the beginning few minutes, we are done with. Okay. Haji. Now, the glycogen storage diseases here right now, sir. Welcome to the glycogen storage diseases. Type 1, 2, 3, 4, 5, 6. You have to remember the name of the glycogen storage disease along with the enzyme deficient. यार सबसे पहले ना मुझे ये बताओ, right from school days we have been learning that glycogen will be stored in our body. It is a storage form of glucose in our body. But can you please tell me where is glycogen stored exactly? Can you please tell me where is glycogen stored exactly? Where is glycogen stored exactly? Yes, look at that answers, guys. Most of the students are answering only liver, but remember glycogen will be stored in liver as well as skeletal muscle. ठीक है ना? ध्यान से. Liver as well as skeletal muscle. Liver as well as skeletal muscle. Now I'll tell you, listen here right now, sir. I'll tell you this one in the easiest manner possible. Listen to me for 2 minutes, guys. Glycogen storage diseases बहुत important है. ध्यान से सुनो. ठीक है जी? Now, do you have glycogen in the liver? Yes, sir. Do you have glycogen in the skeletal muscle? Yes, sir. Glycogen will break down. Yes, sir. Will break down. Yes, sir. What is the name of the enzyme? The name of the enzyme is glycogen phosphorylase. Actually, it's a big अभी के लिए for Glycogen will break down in the presence of which enzyme, sir? Glycogen phosphorylase. Glycogen will break down in the presence of glycogen phosphorylase. Okay, just to differentiate that, remember here, in the liver, it will be like hepatic glycogen phosphorylase, and muscle, yes, I'm just writing here as the muscle glycogen phosphorylase, or you can use the word as myo. Okay. Yes, hepatic glycogen phosphorylase here right now, sir, and this is the muscle glycogen phosphorylase here right now, guys. Okay, ji. Shahid is asking about anatomy. Dikshit is asking about anatomy. A good news to all my dear students, all my dear foreign medical graduates. Yes, I'm going to take the revision session of anatomy tomorrow and day after tomorrow, 27th and 28th. 27th and 28th anatomy wala revision मैं करवाऊंगा आपको, and the link will be given to you. Yes, in the Telegram group. In my Telegram group, you can find the link, guys. Is it okay? So you can join there. It is on YouTube. Tomorrow and tomorrow we are going to bang on with anatomy, guys. ठीक है? तो आज अभी के लिए ना परेशान मत हो. ओके. अरे बाप रे बाप, मैं पढ़ाऊं के गिर जाऊं समझ में नहीं आ रहा है, इतने सारे दिल, वो भी लड़के. ओ माय गॉड, look at that. Rajat Singh is sending so many hearts. Okay. Welcome here, sir. Glycogen is going to break down, and glycogen is going to break down in the presence of the enzyme. What is the name of the enzyme? Glycogen phosphorylase. ठीक है जी? Come on guys. Hello. Everyone here right now, सब लोग यहाँ पर देखो. Everyone along with me right now, guys. Someone is asking Telegram group. It's very simple. Anatomy, Dr. Azam. आ जाओ sir. Welcome here right now. अरे यार anatomy छोड़ो भाई. Just we got like 1 hour. Let us discuss biochemistry here right now. Everyone, please concentrate here right now, sir. Biochemistry. Come on, guys. Okay. Now, muscle. Yes. What is the name of the enzyme? Yes, hepatic glycogen phosphorylase. Carefully. In some unfortunate people, deficiency of hepatic glycogen phosphorylase, it will lead to a disease known as HERS disease. I love the name, HERS disease, and that is type 6. Lovely. And deficiency of muscle glycogen phosphorylase, yes, deficiency of muscle glycogen phosphorylase, type 5 glycogen storage disease. Okay. Internet from my side is absolutely correct here right now, guys. I have checked before the class also. Yes, it is like fantastically going on here. So whenever there is a, like online class, it's like two-way. You should also have like good internet connection. So please try to adjust along with me, cooperate along with me, guys. Let us finish all the topics now. Yes, come on, guys. So how are we going to remember this one? It's like easy. Yes, hepatic, it is HERS. Hepatic HERS, and then muscle glycogen phosphorylase will be deficient in McCall disease. Okay, hepatic will be HERS, and muscle and McCall. H for H, M for M. H for H, M for M. Is it okay? And now, my dear friends, welcome to another one here. Type 5 ho gaya, type 6 ho gaya. Welcome to ABCD. Yes, you all know very well, my dear friends. Yes, you all know very well, my dear friends. A, B, C, D. A for apple, B for ball. Okay, na? Aaj se bhul jao. Forget from today onwards. From today onwards, welcome. A for Anderson's disease. And Anderson's disease will be like type 4. And type 4 Anderson's disease is due to deficiency of branching enzyme. And then, yes, C for Cory's disease. And Cory's disease is nothing but type 3. And type 3, that is for debranching. तो आज से आप लोग कैसे याद रखोगे? A for Anderson, B for branching, C for Cory's, D for debranching. Understanding here right now? So biochemistry is all like a story, like a song, sir. A for Anderson, B for branching, C for Cory's, D for debranching. Is it okay? अगर आप ध्यान से देख रहे हो, if you are looking at my screen very carefully here right now, I'm done with 3 and then 4 and then 5 and then 6 here right now, sir. Easy. So So now welcome here right now, guys. Yes, my dear doctors, look here, look at the screen now. Type 6 HERS disease, it will be hepatic glycogen phosphorylase. Type 5 McCall disease, it is muscle glycogen phosphorylase. And type 4 will be Anderson branching, and type 3 will be debranching, guys. Okay, na? Cory's will be debranching. Lovely, made easy. And this type 2 will be the Pompe's disease. Pompe's disease actually is due to deficiency of acid maltase. And remember, that is actually the enzyme found in the lysosome, sir. So it is more of like lysosome storage disease there. And who doesn't know about the type 1 disease, guys? Type 1 one gig disease, and that deficiency of what's a glucose 6 phosphatase. Glucose 6 phosphatase, guys. Okay. Thank you, Raz. Thank you. Love you. Okay. So these are all the glycogen storage diseases and the enzyme deficiency you are going to remember like this, guys. So best way to do it in this manner, guys. The best way to learn in this manner, guys. Okay. So remember all the names of the diseases and the enzyme deficiencies. Is it okay? And yes, there'll be one more here. Tories will also be there, but that is not required for your MCA exam. वो हम बाद में ना PG में पढ़ेंगे. Okay, these are all required for the MCA exam. Is it okay? So type 1 Von Gigs, type 2 Pompes, type 3 Cories, type 4 Anderson, type 5 McCall, and type 6 will be the Hertz disease, guys. Lovely. Now, after completing all these glycogen storage diseases, now third concept for today completed, and now welcome to the next one, guys. ठीक है ना यार, इसको ना याद रखने के लिए ना order में you have to remember this in order, and to remember that in order, look at that Rajat there. He is giving a very khatarnak, vulgar non-veg mnemonic. Okay, normally nowadays I'm not giving any non-veg mnemonics and all, but look at that. Yes, इसको ना order में याद रखने के लिए Ketan is also telling Von, pumped, Corey, and make her tired. Okay, now so Von, pumped, Corey, and make her tired. Dash. Okay, now B. I did not say anything. Welcome here, right now, sir. Next one. Now, welcome to the next question here, right now, sir. Reduced NADPH is produced in which pathway? Reduced NADPH is produced from which pathway? And now, again, the same thing. With the help of this question, we are going to study about the concept. Which concept we are going to learn now, sir? Which pathway we are going to learn right now? That is your HMP shunt. HMP shunt, it is also known as pentose phosphate pathway. Another important topic, guys. Chalo sir, enjoyment ho gaya. Thoda sa 5 minute na seriously badhte hain. Let us finish this topic right now. What is the full form of HMP? Hexose monophosphate shunt. Hexose monophosphate shunt. It is also known as what, sir? PP pathway. PP stands for pentose phosphate pathway. It is also known as, guys, pentose phosphate pathway. Very good. पहले आराम से ना नाम सीख लो. Learn the name. Now, after learning the name, yes, my dear friends, what is the location of HMP shunt? HMP shunt कहाँ पर होता है? HMP shunt will be taking place in the cytosol, guys. Remember that one. Next is, what is the rate limiting enzyme? Your favorite one. I think you have learned that in your physio, patho, pharma, internal medicine, G6PD. Glucose 6 phosphate dehydrogenase is the rate limiting enzyme. It comes here. है ना? आपने पढ़ा होगा. I think you have learned in your pathology and all. G6PD deficiency will lead to what? Hemolytic anemia and all. And that G6PD comes in this pathway, sir. HMP. Okay. So rate limiting enzyme here will be G6PD, guys. And after that, what is the net gain of ATP? Has been already asked in the MCI exam. Remember, there'll be no ATP produced here right now, sir. Zero. Remember, zero. Okay, na? So HMP shunt, the location will be cytosol. Rate limiting enzyme is G6PD, and the net gain of ATP will be zero here right now. And now, welcome to the most important thing here right now. I can't tell how many star marks you have to write down here. The important products produced in HMP shunt. There are two important products produced in HMP shunt, sir. Number one, NADPH. NADPH. Be very careful in the exam. Your cunning examiner, he will always give you like two options, NADH, NADPH. NADH, NADPH. दोनों देगा आपको. और आपको याद रखना होगा NADPH. HMP shunt, NADPH. HMP shunt, NADPH. And another one will be your ribose 5-phosphate. Remember, ribose 5-phosphate. Okay, guys. So for next 4 days, you have to somehow remember this one. This is what we can do right now. Okay, so what are the two important products produced here? One is your NADPH, another one is the ribose 5-phosphate. And they have got like many importance, guys. Ribose 5-phosphate will be necessary for the formation of your DNA and RNA. Okay, that is required actually for purine synthesis. Fine. NADPH, I'll tell you the logic. Remember like this, guys. It is required for fatty acid synthesis. Arey yaar, ye batao. Fatty acid belongs to which which group? Fatty acid belongs to what? Lipids, right? So if it is required for fatty acid synthesis, can I write down here as lipid synthesis? Yes. If it is writing as lipid here right now, sir, can I write down as cholesterol synthesis? Yes. And if cholesterol is forming here, can I write down here as steroid hormone synthesis? Of course, it is also required for steroid hormone synthesis. Okay, now, so I request all my students to just remember one single word there, fatty acid synthesis. Everything else will follow. So NADPH is required for fatty acid synthesis, also for lipid synthesis, also for cholesterol synthesis and steroid hormone synthesis. Next one. NADPH is required in RBC. Why? It is required inside the RBC to remove the free radicals. And NADPH is also required inside the neutrophils. And inside the neutrophils, my dear friends, it is required for killing the bacteria. I think you have learned that in pathology. You will are very good in pathology, you know. Killing the bacteria will be by two methods: oxygen dependent, oxygen independent. Yes, Amit, you are absolutely right there. It is required for oxygen dependent killing of the bacteria. Lovely. So it is like, you know, like tag, 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 like, you know, A, B, C, D, 4 points that you have to remember for your MCA exam. Done, sir. Okay. So promise me in the morning of 31st August. Come on. HMP shunt is also known as pentose phosphate pathway. Location, cytosol. Rate limiting enzyme, G6PD. Net gain of ADP, 0. Important product, there'll be two important products obtained, guys. Lovely. You are going to learn this or revise this and go to the exam, guys. Lovely. Welcome back. Reduced NADPH is produced from which pathway? It is by HMP shunt. Done, done, done. When comes it? Next one. Fructose intolerance is due to deficiency of what, guys? Okay. Fructose intolerance is due to deficiency of what? Let's see what is the enzyme deficient. And now, my dear doctors, listen here. Whenever carbohydrates are undergoing metabolism, come on, sir, dhyan se, full concentration here. 200% concentration here, sir. Everyone here. Forget about everything in your life, especially forget about girlfriend and boyfriend in your life right now, sir. Okay, now, bhul jao. Welcome here. Whenever the carbohydrates are undergoing metabolism, or sorry, digestion, we are going to form glucose, fructose, and galactose. Glucose, fructose, and galactose. Okay, now. Glucose, fructose, and galactose. Okay. And out of them, my dear friends, welcome to the fructose and galactose. And you have to remember like two things right now, sir. Only two things right now. Welcome here right now, guys. See, the metabolic disorders you got to remember. Okay. And these two are the metabolic disorders associated with the fructose metabolism. And this one is for the galactose metabolism. Okay, now. Now welcome here. Come on, guys. No cross comments. सब लोग यहाँ पर concentrate करो. ठीक है ना? है ना? एंजॉयमेंट के टाइम पर एंजॉयमेंट, पढ़ाई के टाइम पर पढ़ाई. ठीक है ना? नो गड़बड़ी हियर. अब वो मैं देख रहा हूँ वहां पर ना, एक बोल रहा है, एक बोल रही है. सो देयर इज नो क्रॉस कमेंटिंग हियर, सर. यस, कॉन्सेंट्रेट हियर, राइट. वेरी वेरी लेस टाइम लेफ्ट आउट. इट्स ऑल अ मैटर ऑफ लाइक 17 मार्क्स. 17 मार्क्स, गाइस. ठीक है. सो वेलकम हियर राइट नाउ, सर. एंजॉयमेंट करेंगे. ठीक है ना? सो आवर मोटो इन द लाइफ इज द सेम थिंग, है ना? वर्क हार्ड एंड द पार्टी हार्डर. ठीक है. वेलकम हियर राइट नाउ, सर. Fructose metabolism, full concentration. Fructose metabolism, two things you have to learn here right now. Number one, essential fructosuria. It will occur due to deficiency of which enzyme? The name of the enzyme is fructokinase. Listen very clearly. First of all, I think there's some disturbance. I am telling fructokinase. Is it okay? Essential fructosuria is due to deficiency of which enzyme? Fructokinase. More than this is more important, guys. Fructose intolerance. Fructose intolerance will occur due to deficiency of Aldolase B. Fructose intolerance is due to deficiency of Aldolase B. B is also important there. I think you have learned that in pediatrics also. Lovely. So in fructose metabolism, no need to learn anything else, guys. The only two things that you have to learn here is essential fructosuria and fructose intolerance. Out of these two also, fructose intolerance will be more important, guys. That is due to deficiency of what? It's due to deficiency of Aldolase B. Lovely. Done. Now welcome to galactosemia. There's a big enzyme name here right now, sir. So learn it properly. Aram se along with me. Okay. The name of the enzyme will be galactose 1-phosphate. Okay. Galactose 1-phosphate, and that will be uridyl transferase. Galactose 1-phosphate uridyl transferase. Okay, now. So during our regular classes, guys, we'll be having a lot of time that time. I'll be teaching like entire, you know, the metabolism of that, how to make the name of the enzyme. But abhi ke liye, do something, remember this and go to the exam, guys. Okay. Galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase. Yes. Lovely, guys. Okay. Harivardhan is telling there, Gali put it seems. Wow, lovely. I love you all. You have got like all sorts of, you know, khurafati ideas. Yes, Gali put. Okay. Galactose 1-phosphate uridyl transferase. And due to that, what is going to happen? Galactosemia. The galactose level will increase in the blood. And that galactose will be actually going to your, eyes. And I think you have learned that in ophthalmology, my dear friends, galactosemia की वजह से क्या होगा? It will lead to cataract, and that will be your oil drop cataract. चलो सर, तो हमने one more point we have revised from the ophthalmology also. क्या होगा यहाँ पर? Oil drop cataract होगा. Lovely, love you all. Look at the answers with confidence. Yes, we are going to clear the exam, है ना? Essential fructose urea enzyme deficiency. चलो सर फिर से रिवाइज करते हैं. You also tell loud and clearly. I'll also tell loud and clearly, है ना? तुम भी पागल, मैं भी पागल. ठीक है ना? Essential fructose urea fructokinase. Fructose intolerance will be due to deficiency of Aldolase B, and galactosemia will occur due to deficiency of galactose 1 phosphate uridyl transferase. Galactose 1 phosphate uridyl transferase. An increased level of galactose level in the blood, galactosemia, will lead to a cataract, and that is nothing but your oil drop cataract. Oil drop cataract. Done. So these are the 3 points that you have to remember. Yes, please, guys, mark my words. I hope you're writing there, guys. These are the only 3 points you have to remember in the galactose and fructose metabolism. Abhi ke liye. For rapid revision, I can help you this much. ठीक है ना? There'll be so many students like sir हमने ना बायोकेमिस्ट्री जिंदगी में नहीं पढ़ा है यार. अभी at the last moment, if you want to do something, if you want to get the marks in biochemistry, these are the points you have to remember, you have to learn, guys. ठीक है? So welcome back here right now. Fructose intolerance is due to deficiency of Aldolase B or Aldolase A, guys. Fructose intolerance, remember Aldolase B. Lovely. Done, done, done. Next one, sir. After that, can you answer this question here right now, sir? Enzyme deficiency, which is the most commonly responsible for galactosemia. Galactosemia के लिए कौन सा वाला enzyme responsible होगा? Answer in the poll now. Answer in the poll now, everyone, sir. Your time starts now. Up. आपका समय शुरू होता है now. क्या बात है, क्या बात है. Look at that confident answers from all my students here right now, sir. But still I'm getting the percentage here as 96%. भाई ये 4% कौन से वाले लोग हैं? अभी अभी मैंने पढ़ाया था आपको गैलेक्टोसीमिया. अरे बाप रे, we got 95% result. I don't know who is this 5%. I just not taught you galactosemia. It's due to galactose 1 phosphate uridyl transferase. Okay, चलिए आगे बढ़ते हैं. Let's go ahead. Now the next concept. तो अगर आपने ना ध्यान से देखा, if you are observing properly, glycolysis done, Krebs cycle done, glycogen storage diseases done, HMP shunt done, galactose metabolism and the fructose metabolism done, guys. Understanding? So I have revised like almost the entire carbohydrate metabolism, sir. अगर आप मेरी classes ना properly follow कर रहे हो, then there will be a lot of benefit out of that, guys. Entire carbohydrate done. And now, yesterday proteins done, today the carbohydrates are done, and now welcome to the lipids topic. Let us learn what to learn in the lipids, guys. Welcome. Welcome, guys. What do you have to learn in the lipids here right now? The most essential fatty acid. Oh, ho, 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 ho. Write down in your books there. Okay. Definite question on 31st August 2020. आ चुका है, सर, print हो गया है, ठीक है ना? Look here. Come on, बोलो बच्चों, fill in the blank here right now. What is a lipid made up of? Lipid is made up of 2 things. Number 1, fatty acid, and that will combining along with the alcohol, and most commonly it will be like glycerol. Okay, ठीक है. So what is a lipid here right now, sir? Lipid is an ester of fatty acid and glycerol. It's an ester of fatty acid and glycerol. Is it okay? तो जिंदगी में याद रख लो, throughout all your 19 subjects, anywhere you come across a lipid, you have to remember these 2 words here right now: fatty acid, glycerol, fatty acid, glycerol. And now welcome to fatty acid. Classification of fatty acid. In the classification of fatty acid, my dear friends, there is one classification. Yes. The classification of fatty acid, the one classification is that based on nutritional not produced in my body and I essentially require in diet. जो मेरी बॉडी में प्रोड्यूस नहीं होगा, इसीलिए मुझे essentially diet में लेना होगा. Essential fatty acids, there are 3 of them: linoleic acid, linolenic acid, and arachidonic acid. Linoleic acid and linolenic acid and arachidonic acid, guys. These are the 3 essential fatty acids you have to remember here. And now, my dear friends, what do you have to remember in this one? Point number 1, you have to remember their nomenclature. Okay, I'll tell you the nomenclature of only the first one, guys. I think remaining you can handle. Welcome. Linoleic acid, remember the nomenclature here is like, 18:2, 9, 12. Sir, what should you understand out of this one? सबसे पहला जो number होगा, the first number is representing the number of carbons here right now. Swaha. The second number is representing you the number of double bonds here right now, guys. Swaha. समझ में आ रहे हैं? Number of carbons in linoleic acid, 18 carbons. How many double bonds? 2 double bonds. The moment you know that there are 2 double bonds, you should know the position. Which position? Double bond position. The position. And write down completely, my dear friends, position of what? Double bond. Is it okay? Position of double bond is important. Understanding? So 18:2, 9, 12. 18 number of carbons, 18 carbons. And then 2 number of double bonds. Where are they present? 9 carbon and the 12 carbon, guys. Done. Swaha. Next one, sir. Apart from this, nomenclature here right now, you also have got the omega. And I'll tell you one simple trick here right now for the last moment, guys. First number minus the last number. First number minus the last number. Okay, now come to my regular classes. I'll teach you exactly wo kaise hota hai. For now, you remember 18 minus like 12, it will be 6. So this will be like omega-6 fatty acid, guys. Linoleic will be the omega-6 fatty acid. Omega-6 fatty acid here right now, sir. Done, done, done. I think remaining you can manage. It's just, memorizing there, guys. It's a factual question. The remaining nomenclature, I hope you'll be able to manage. But important concept here, what to remember? Nomenclature here right now, guys. Done. And then question number 2, MCQ number 2 possible here. These 3 are essential fatty acids. Out of that, which is the most essential? Linoleic acid is the most essential fatty acid. Linoleic acid is the most essential fatty acid, sir. Done, done, done. अब यहां से सुनो ध्यान से, guys. There was with pharmacology. अब मैं थोड़ा सा ना आपकी ना pharmacology में उंगली करता हूं. सुनो ध्यान से. Do you remember pharmacology? Are you all done with the pharmacology? Yes. Are you all done with the pharmacology, guys? Come on. Have you learned about NSAIDs? Have you learned about the non-steroidal anti-inflammatory drugs, NSAIDs? And there, I think you have learned about the arachidonic acid. Arachidonic acid will undergo metabolism, COX and LOX. Okay, na? COX pathway and the LOX pathway. COX and LOX pathway. ठीक है ना? So don't take me wrongly, it is cyclooxygenase and the lipooxygenase. Cyclooxygenase and the lipooxygenase. और वहां पर क्या आता है सर? Arachidonic acid आता है, ठीक है ना? And it is nothing but one of the essential fatty acid. समझ में आ रहे हैं? तो you have to correlate these two here right now. Okay, arachidonic acid is an essential fatty acid, and remember that in the pharmacology part, guys. Done, done, done. So welcome back to this question here again. The most essential fatty acid will be your linoleic acid. Linoleic acid will be the most essential, guys. So please mark there in your books. Very important one, the star mark. Definite question in your exam, guys. Next question right now. Ketone bodies cannot be utilized by which of the following organ? My dear friends, yes, remember here right now, the ketone bodies cannot be utilized by which of the following organ? So my concept here is about the ketone bodies. Chalo, chalo. What are the 3-4 points to be remembered? Bolo bachcho. What are the 3 ketone bodies? Number 1, acetoacetate. Write down the name and repeat the name. Number 2, acetone. And remember the number 3, that is beta hydroxybutyrate, guys. Beta hydroxybutyrate. These 3 will be the ketone bodies. These 3 are the ketone bodies. First of all, learn the 3 ketone bodies. I repeat again: acetoacetate, acetone, and the beta hydroxybutyrate, guys. Acetoacetate, acetone, and beta hydroxybutyrate. These 3 are the ketone bodies. And now, what are the points to be remembered here right now, my dear friends? Write down there: ketogenesis, formation of the ketone bodies. Where it will take place? In the liver. Okay, where inside the liver? Remember, the ketogenesis will take place in mitochondria. Already this has been asked in your exams. MC exam mein pucha gaya hai. Keep in mind, sir. It will be taking place in the mitochondria of the liver cells. Done. And one more question here right now, my dear friends. Tell me, out of the 3 ketone bodies, which is the primary ketone body? Which one will be the primary ketone body? Yes, the primary ketone body will be the first one here right now. That is your acetoacetate will be the primary ketone body. Okay. I think you all know, yes, these are the points to remember here right now. And now, my dear friends, I think you all know that ketone bodies are a very good source of energy, especially during the time of starvation. fasting, starvation. They are very good source of energy. Almost all the organs, even like brain, also utilizes ketone bodies. But the question is, which organ cannot utilize the ketone bodies? Yes, that is your RBCs here right now, sir. Your heart, your brain, your skeletal muscle, all of them can utilize. But remember RBC, and even add there one more thing, even liver also cannot utilize the ketone bodies, guys. So everything regarding ketone bodies, done, 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 sir. Kalas. समझ आ रहा है? So remember the names of the ketone bodies. Where is the ketogenesis taking place? Liver. And then remember the mitochondria. And remember where it is not going to take place in the RBC and the liver, guys. Done. Now the next concept, guys. So I'm completing like every question possible. Now this question I have selected here. This question, guys, I have selected here. Yes, to teach you about the number of ATPs. They can ask you a question based on the number of ATPs in each and every pathway. So let us see which pathways you have to learn for number of ATPs. Welcome. One, you have to remember aerobic glycolysis. How many number of ATPs? What is the net gain? There will be 7 ATPs produced in aerobic glycolysis. Anaerobic glycolysis, there will be 2 ATPs, net gain of ATPs. In the Krebs cycle, you have to remember, like, net gain will be like 10 ATPs, guys. And yes, in the beta oxidation of fatty acid, that will be 106 ATPs. And human beings like you and me, remember which fatty acid is undergoing metabolism, sir? Which fatty acid? That will be your palmitic acid. Okay, that will be palmitic acid. So if it is palmitic acid, it will be 106. So my dear friends, this question is about the number of ATPs. So I have, made this entire series, all like which concepts to be touched and all. And then Krebs cycle 10 and metabolism of palmitic acid, beta oxidation of the fatty acid, it will be 10 limiting enzymes. And now, since yesterday I have been teaching you about the rate limiting enzymes. And since yesterday I have been teaching you about the rate limiting enzymes. Okay, rate limiting enzymes. So now, my dear friends, what we'll do is we'll summarize all the rate limiting enzymes at one place here right now. So welcome. Okay, come on. Glycolysis, today I taught you in the very beginning. I told you already in glycolysis, the rate limiting enzyme is, yes, PFK1. We already know about this one. Okay, no need to worry. Krebs cycle, yes, we already know it is isocitrate dehydrogenase. We have learned that one, guys. Okay. And HMP shunt, already I told you it will be G6PD. Already we have learned that one. And now welcome to the remaining pathways. First of all, welcome to this glycogen wala, sir. Glycogenesis, formation of glycogen. For the next 3-4 days, remember, we are forming glycogen, forming synthase, glycogen synthase, and then glycogenolysis breakdown. मैंने आपको already बताया है. What is the name of the enzyme? Glycogen phosphorylase, guys. In the liver, hepatic, and in the muscle, it will be nothing but your, yes, the muscle glycogen phosphorylase. Okay. So glycogenesis, formation of glycogen synthase, phosphorylase, guys. Already I told you Krebs cycle and HMP shunt. Now please remember these two. I told you how to remember. And now the only one which is very much important also to remember here right now, guys, gluconeogenesis, formation of glycogen from, formation of glucose from the non-carbohydrate sources. Formation of glucose from non-carbohydrate sources. I think this is the only one that you have to learn here right now. That is fructose 1,6 bisphosphatase. Fructose 1,6 bisphosphatase, guys. Done. तो अगर आप ध्यान से देखोगे, इट इज नॉट एट ऑल डिफिकल्ट गाइस. ग्लाइकोलाइसिस हमको ऑलरेडी पता है, हम लोग बचपन से पढ़ रहे हैं फ्रॉम स्कूल डेज. Krebs cycle we already know. G6PD, HMP shunt we already know. Glycogen, I made it easy for you right now. Breakdown, phosphorylase, and remember formation, synthase. And this is the only one you have to remember. Fructose 1,6 bisphosphatase. These are all the rate limiting enzyme. Examiner can ask the question based on rate limiting enzyme. I'm expecting one question based on rate limiting enzyme, guys. Is it okay? So that's important here right now. Done. So therefore, welcome back here right now, guys. What is the question? All of the following are rate limiting enzyme except one. Phosphofructokinase, yes, it is a rate limiting enzyme. Glycogen synthase, rate limiting enzyme. Glycogenesis, glycogen phosphorylase, yes, rate limiting enzyme. Glycogenolysis, but not the glycogen phosphatase, guys. Done, done, done. So another thing done. Questions based on number of ATP are done. Questions based on the rate limiting enzyme has been done. Now let me tell you the next one, guys. Which of the following is not a rate limiting enzyme? Sir, आप ये अब आपने दोबारा क्यों दिया? Why did you give this again? Look at the options there, guys, carefully. HMG CoA reductase is the rate limiting enzyme. Yes, it's a rate limiting enzyme. Of which pathway? Of, yes, cholesterol synthesis. Synthesis of cholesterol. Very good. Very important there, guys. Yes, cholesterol synthesis. It will also come in pharmacology. Yes, very good, Inderjit Singh. That will be inhibited by statin group of drugs. Phosphofructokinase. It is also a rate limiting enzyme of, yes, glycolysis. PFK1. It is the rate limiting enzyme of glycolysis. Okay. And then acetyl CoA carboxylase. It is also the rate limiting enzyme of, yes, fatty acid synthesis. So I gave you this another question. In the first question, it was all the rate limiting enzyme of carbohydrates, and now it is about the lipids, guys. Okay, now fatty acid synthesis, and this is not the rate limiting enzyme. So all the rate limiting enzymes are completed. We have revised them. Is it okay? And now welcome to the next one. If the content of adenine is 15%, then what is the amount of guanine in the DNA according to Chargaff's rule? बोलो बच्चों, चार्गस भाई साहब ने क्या कहा था? What is Chargaff's rule? Chargaff's rule is adenine loves to bind with thymine double bond, and guanine loves to bind with cytosine with the triple bond. Guys. Adenine with thymine, guanine with cytosine. And I'll tell you how to calculate this one. Don't panic. It is the easiest way to calculate, guys. I will tell you here. Welcome here right now, sir. How to calculate this one, sir? No need of any formula, nothing. Look at the screen right now. Welcome. Adenine is how much? 15%. Okay. Then thymine will be how much? 15%. 15 plus 15 will be how much? 30%. Okay, G. If it is 30%, guanine and cytosine, this will be definitely the remaining 70%. Why? Because I want my DNA to be 100%. Perfect. Okay, perfect DNA. If these two are 70%, then cytosine will be 35 and guanine will be 35%. What is asked in the exam, sir? Guanine. Guanine is how much? 35%. हो गया. It's so simple. No need of any complicated formula. कुछ नहीं चाहिए. Chargas भाई साहब, Chargas rule. Adenine loves to bind with thymine. Guanine loves to bind with cytosine. Done. This is how you are going to handle. Already these type of questions are given in the MCI exams. Lovely. No need of formula, nothing, guys. Next one. What is a nucleotide? I hope you all know that our DNA will be actually, double stranded. Our DNA will be having like one strand here like this, and there'll be another strand here right now, sir. And what is this strand made up of? Polymer of nucleotide. It is made up of many, many nucleotides. And now we want to learn in depth, sir, what is that each nucleotide made up of? I repeat again, every student knows that DNA double stranded. Each strand is a polymer of nucleotide. But yaar, nucleotide kya hai? What is a nucleotide? Write it like this. Likho sir mere saath mein. Nucleotide is made up of three things, sir. It is made up of a sugar molecule. There will be a sugar molecule along with phosphate group and along with the nitrogenous bases. I hope you all know the nitrogenous bases very well. The purines and pyrimidines, guys. Adenine, guanine, cytosine, and thymine. So, the sugar molecule with phosphate and the nitrogenous base. Very good, Aarti. Very nice. Sugar, phosphate, and nitrogenous base. And now, my dear friends, what is a nucleoside? A nucleoside is only a sugar molecule along with the nitrogenous base. That's all. If you remember these two things here right now, you will never make a mistake in these type of questions, guys. So a nucleotide will be sugar, phosphate, and nitrogenous base, and a nucleoside will be only sugar along with the nitrogenous base. There will be no phosphate group. So yaar batao yahan par, what is a nucleotide? It is nothing but the nucleoside plus the phosphate group. Lovely. Okay, nucleoside plus along with the phosphate group. Like, chaliye sir, next one. Which enzyme is deficient in the LESCH NYHAN syndrome? The enzyme deficient in the LESCH NYHAN syndrome is, come on guys, answer. Can you please answer me right now? Let's take a poll here right now. The enzyme deficient in the LESCH NYHAN syndrome. Come on, my dear doctors. Come on. Lovely, lovely, lovely. That's really nice, guys. Okay, now. So 81% of the students have answered correctly here right now. Welcome here. Which enzyme is deficient in LESCH NYHAN syndrome? It will be HGPRT. HGPRT. Okay, now hypoxanthine. Okay, hypoxanthine guanine phosphoribosyl transferase. hypoxanthine guanine phosphoriboside transferase. This comes in purine synthesis, salvage pathway of purine synthesis में आएगा सर. ठीक है, done. In the LESCH NYHAN syndrome, yes, in the LESCH NYHAN syndrome, the most important feature in that one will be the self-mutilation. The person will be harming him own self. Okay, now. So in LESCH NYHAN syndrome, what is the important feature there, guys? The characteristic feature, self-mutilation. So if you find this kind of question, okay, an image-based question, okay, self-mutilation, the person will be harming himself. So biting the fingers, or chewing the fingers and chewing the lips there, all these fingers will be damaged and the lips will be damaged and all, guys. So the moment you see the option there as self-mutilation, go for LESCH NYHAN syndrome. Apart from that, there will be neurological disorder. The person will be suffering with gout and everything, guys. Okay, so this, yes, gout also will be there. Yes, Ravi Nayak. Okay, na? So the most important thing, the keyword how to identify in the exam will be the self-mutilation. Okay, LESCH NYHAN syndrome. Okay, next one, guys. Acetyl CoA can be converted into all of the following except one. Okay, यहाँ देखो ध्यान से. This is like a very simple question here right now, my dear friends. Remember, my dear friend, acetyl CoA is the starting substance for the synthesis of fatty acid. Acetyl CoA is going to form the fatty acid. Acetyl CoA is the starting substance for the synthesis of cholesterol. And acetyl CoA is the starting substance for the synthesis of ketone bodies also, guys. Understanding? So, yes, again and again, I'm telling you guys so that, you can just go back and refer. After the class today, don't even learn the entire pathway. Just जाकर ना notes को एक बार refer करो sir. Just refer to the notes there. In the notes, yes, my dear friends, you can see there, fatty acid is going to begin with where, sir? Fatty acid synthesis, acetyl CoA. Cholesterol synthesis begins with acetyl. Therefore, my dear friends, remember, it is going to form all of them except the glucose. Remember, it cannot get converted into glucose, guys. It can form everything here right now. So I'm just trying to cover all the many points in one question itself. So instead of asking you separate like one question there, guys, कि what is the starting substance of cholesterol, ketone bodies, ना, एक ही question में मैंने सब कुछ include कर दिया है. I've included like everything in one question. At least you should be able to remember this one. Is it okay? Next one, sir. All are derived from cholesterol. 2019 MCI exam question. 2019 MCI exam question, guys. Come on, my dear doctors. Cholesterol is undergoing degradation to form what? Cholesterol is going to form what, sir? All the steroid hormones. Yes. Each and every steroid hormone will be derived from where only, sir? Cholesterol. I think you have learned this in the physiology. In the endocrine physiology, the physiology faculty will teach you there, guys. Yes. All your steroid hormones, okay, in your body. Mineralocorticoid, glucocorticoid, testosterone, estrogen, anything, any steroid hormones, ultimately it's formed from where? Cholesterol. Okay, that's really good. I love cholesterol. I need cholesterol. It is forming testosterone. Cholesterol is the one which is going to form vitamin D. I don't know like how you people remember. मैं तो ऐसे याद रखता हूँ भाई, cholesterol बहुत important है. Cholesterol हमको चाहिए. I need cholesterol. Okay ना? It is going to form. And vitamin D is required for what, sir? Bones. Yes, I need cholesterol. And remember, cholesterol is the one which is going to form all your, bile acids also. And bile acids are nothing but they're going to become your in the form of salt, guys. Okay, na? So bile acids and the vitamin D and the steroid hormones, all this will be formed from cholesterol. It's a very important substance. पता नहीं लोग क्यों those things, coronary artery diseases and all. Nah, cholesterol is very good, bhai. It's very good. Is it okay? So it's going to form steroid hormone, the vitamin D, and bile acid. I think at least you'll remember like this. Hai na? Vitamin D, yes. Bile salts, yes. Steroids, yes. But not the bile pigment. But not the bile pigment, guys. Okay, not the bile pigment, guys. ठीक है ना? Bile pigment क्या होता है actually, sir? Bile pigment is the one which is nothing but your bilirubin, and remember it is formed from the heme. So have a clarity there. So इसी के लिए मैंने question include किया है. That is the reason I have included this question. In the exam, 19 subjects will be there in your mind, guys. And out of that 19 subjects, guys, full of tension. And in the exam, bile acid, bile salt. I have seen students getting confused there. Don't get confused, guys. Okay, bile acids and bile pigment अलग होता है. Next one. Now, I think almost everything covered here right now, guys. All the rate limiting enzymes. All the rate limiting enzymes are done. And after that, we are done with all the number of ATPs produced. all the pathways, carbohydrate, essential fatty acid, classification of the fatty acid, everything is done. And now from here onwards, I have just included at last. Wantedly, I just kept it at last, sir. Why? Because I know every student will be perfect in vitamins. Okay, now listen my words with all your ears open. Vitamins topic is very much important. I have observed the question paper very closely from years together, guys. FMG, every exam minimum two questions will come from vitamins, and maximum they have even asked up to 5 questions of the vitamins also, guys. Minimum 2 marks. Vitamins padkar jana zaruri hai. Is it okay? Come on, start. Burning food syndrome is due to deficiency of which vitamin here right now, sir? Yes, that is your which one? Pantothenic acid, vitamin B5. Lovely. Okay. I'm not going in detail about the vitamins right now. Almost like our time is done also. And apart from that, vitamins आपको पता ही होगा. Next one. Biotin is a cofactor. Yes, for which enzyme? Remember, it is a cofactor for carboxylases. I'll show you one slide here right now. Remember, biotin is actually acting like a coenzyme. Biotin acts like cofactor or coenzyme, coenzyme there for how many enzymes, guys? At least you have to remember these three here right now. Pyruvate carboxylase, it comes in gluconeogenesis, okay, pathway. Propionyl CoA carboxylase, it comes in propionic acid pathway, vomit pathway, propionic acid pathway करके होता है. And after that, acetyl CoA carboxylase, already I told you, this enzyme is an enzyme of fatty acid synthesis, guys. Are you understanding the point here right now? So carbohydrate metabolism, protein metabolism, and this comes in lipid metabolism. But the beauty here, carboxylase, carboxylase, carboxylase. What is required? Biotin. So biotin acts as a cofactor, a coenzyme. Yes. For which enzyme, guys? For all your carboxylases. Important. Next one here. Another name for vitamin K2. I have included this question for you to recall. Yes, your examiner is asking like simple question. They're even asking about the names of the vitamin, guys. So what is the name of the vitamin K2, sir? Phylloquinone, menadione, menaquinone, and calcitriol. Yes, it is menaquinone. Remember the names of the vitamins. Deficiencies of the vitamins and the names of the vitamins, guys. Next one. Which of the following is an insulin dependent transporter? Come on, guys. Which of the following is an insulin dependent transporter? Yes, that is nothing but your glucose transporter number 4. That is insulin dependent transporter. And remember, glucose transporter number 4 will be present on what? It will be present on skeletal muscle. Okay. And remember, it is also present on the adipocytes also, guys. These two are the important locations you have to remember. And on the surface of skeletal muscle and on the surface of adipocytes, the number of glucose transporters are increased under the influence of which hormone? The hormone is your insulin, guys. That is the reason why it is known as insulin dependent. Insulin dependent glucose transporter. Uska number बढ़ता है, ठीक है ना? And Farooq is asking about the GLUT2. Remember GLUT2, Farooq, it will be present on the liver. And GLUT2 will be present on the beta cells of pancreas. And it is also present on the kidney also, guys. And Farooq, don't get confused. It is not like insulin dependent. It is required for the release of insulin, secretion of insulin. So GLUT2 on the beta cell. Beta cells will secrete insulin. उसके लिए चाहिए. तो ये दोनों question में ना confuse मत होना. तो Farooq has raised a very good question here right now, guys. Insulin dependent. I repeat again, guys. Insulin dependent, GLUT4, and insulin secretion. Which one is required, guys? The GLUT2. GLUT2, guys. Lovely. And the GLUT1 will be present on your brain, and it is present on the RBCs. Okay, now even recently asked question in the MCI exam, it is also present on the placenta, guys. Even GLUT1 as well as GLUT3, both of them are found in your brain, RBC, placenta, and all. GLUT2 will be present on the liver, beta cells of pancreas and kidney, and the GLUT4 on the skeletal muscle and adipocyte. And remember, it is the one which is insulin dependent transporter, guys. Insulin dependent transporter, my dear friends. Okay, so that was about the rapid revision session of the biochemistry, guys. Rapid revision session of the biochemistry. Yesterday like 1 hour and today like 1 hour, and we have completed proteins, we have completed carbohydrates, we have completed the lipids, electron transport chain, and the enzymes topic. Almost all the topics I have completed everything, guys. Okay, at least before 1 day before the exam, look at this one and go to the exam. And apart from that, guys, a small LMR, last minute revision of the biochemistry is there, just 3 pages. Okay, now if you need that, I'll just forward you that in the Telegram group, guys, and you can take there just 3 pages, keep that with you. All these notes will be there in the 3 pages. Okay, now, and in that every point is mentioned, and aram say you can just revise it and go to the exam, and that will give you like maximum number of marks out of 17. So I think I have helped you with this one. I think these 2 hours before the exam were very much useful to you people, guys. So thank you all, thank you all, thank you all. So prepare in all these things, guys. All the best for your exam. ठीक है ना. So I think I was of a little bit help in your journey, guys. तो आपकी जो important ये journey है FMG exam को clear करना है तो आराम से करिए sir. It is not all a difficult exam. Out of 300 we have to get like 150. Okay. 150 marks is just like, okay na. So all the best. Stay safe, stay blessed, take care, and I'm always there for you guys. I want to see all your responses on the day of the exam. So after the day of the exam, come back and give me all your reviews about this session, guys. So thank you all. And one more important thing is follow me on the Unacademy, guys. If you follow me on the Unacademy, you'll be getting all the updates regarding my all this type of classes, guys. I'll be conducting like many of the free classes. So follow me on the Unacademy, and you'll be getting all the updates. So thank you all. Take care. All the best, guys.